Okay, I want to talk about another metabolic pathway, and that is gluconeogenesis. Gluconeogenesis is essentially the reverse of glycolysis, but not quite. So don't get in the habit of saying it is the reverse because it's not exactly the reverse. Okay, but basically, it's the same sort of process. We're starting with pyruvate, which was the product of glycolysis, and we're making glucose, which was the initial substrate of glycolysis. Okay, so we're taking pyruvate, a 3-carbon molecule, and eventually making a 6-carbon glucose molecule. Okay, and that's essentially what gluconeogenesis is. The goal is to make glucose. Okay, so let's start looking at the steps real quick, the basic steps taking us through the gluconeogenesis process. It's an 8-step process, and several of the enzymes are identical to those found in glycolysis, but not all of them are, because if you recall, there were some reactions in glycolysis, reactions 1, reactions 3, so the phosphorylation of glucose or the production of fructose 1,6-bisphosphate, and reaction 10, where we actually made pyruvate and ATP, all had significant energy or made ATP. Okay, they made ATP. So they were very, had a lot of free energy and whatnot. So what that does is creates energy barriers for gluconeogenesis. So it's not going to be as easy to get over those steps. So the first thing I want to point out is that steps 1 and 2 here in gluconeogenesis are simply steps that are needed to overcome step 10, which was catalyzed by pyruvate kinase in glycolysis. Okay, so to overcome that step, if you recall, it was phosphoenolpyruvate to pyruvate to making pyruvate, okay, and phosphorylating ADP. Okay, but look at what has to happen here. So I start with pyruvate, and another thing I want to point out is that there's 2 molecules of everything here. So there's 2 pyruvates here, there's 2 oxaloacetates, and everything's doubled all the way down. Okay, and that's just a result of what happened in glycolysis. Okay, so pyruvate plus ATP plus carbon dioxide plus water gives me a new molecule, a new 4-carbon molecule known as oxaloacetate. Okay, plus ADP plus PI, inorganic phosphate, plus 2 hydrogen ions. Okay, and the enzyme that's catalyzing that reaction is known as pyruvate carboxylase, and that's really important because that's, again, think about it, we need energy here. Look, we're using ATP in order to make this, we're coupling this reaction to ATP hydrolysis to make this favorable, okay, to overcome the energy barrier. And if that tells you anything, like I said before, anywhere where you're going to need energy in a metabolic pathway where it's going to require ATP hydrolysis, some form of energy input, you can almost bet that there's going to be regulation at that step, okay? And you'd be right in thinking that here, okay? So biotin is also assists in the addition of CO2, so that's like a cofactor that's going along, or coenzyme that's going along with pyruvate carboxylase to help in the process. Okay, and the second step is, again, it requires 2 steps to overcome this. If you look at the product we're making in step 2, it's phosphoenolpyruvate. And remember I said phosphoenolpyruvate is a high energy molecule, okay, that's produced in glycolysis. And in order to produce that high energy molecule, again, we need GTP. So if we're using GTP, which is another triphosphate similar to ATP, that means, it's requiring an input of energy, so there's an energy barrier to overcome there. And that's going to give us the phosphoenolpyruvate, GDP, and CO2. Okay? The enzyme is known as PEPCK, and I believe that stands for phosphoenolpyruvate carboxylase kinase. Okay? So that's a mouthful. It's easier to say PEPCK. Now, moving on to step 3, this is where things start to look a little bit more like glycolysis. Phosphoenolpyruvate is converted into 3-phosphoglycerate, okay? And in step 4, 3-phosphoglycerate becomes 1,3-bisphosphoglycerate, and the enzyme there is phosphoglycerate kinase. Step 5, we have 1,3-bisphosphoglycerate plus NADH plus H plus gives us fructose 1,6-bisphosphate, remember the first unique product of glycolysis, plus NAD plus. So take a look at this. Look, we need NADH. And under what conditions is NADH found? Well, NADH is only found under energy favorable conditions. So when the cell has a lot of energy, there's a lot of ATP, everything's going well, then you have excess NADH, okay, which is a reduced electron carrier here in this case. And that's going to produce fructose 1,6-bisphosphate. So I have a little note here that says NADH is required. We will have NADH when the cell has plenty of energy, okay, and the cell will only make glucose when there is an abundance of energy. So this is not a catabolic process like glycolysis. That was a catabolic process where we're breaking something down, okay. This is an anabolic process. We're building a larger 6-carbon molecule from 2 smaller 3-carbon pyruvate molecules. So this is going to take more energy, okay, plain and simple. Anytime you're building something, it always requires more energy. It's easier to break things down and generate energy from the breakdown of the molecules rather than to build them up. So that's why I put that in there, and I explained why we see NADH or why NADH is involved. So fructose 1,6-bisphosphate gives us fructose 6-phosphate plus inorganic phosphate. Okay, so what's happening here is a phosphate is being clipped off, and the enzyme that's doing that is known as fructose 1,6-bisphosphatase, another important enzyme. So I'll point it out now. This is going to be some regulation going on here, so you'll want to remember fructose 1,6-bisphosphatase. Step 8 is glucose 6-phosphate. Goes over to forms glucose. Well, I shouldn't have skipped this step. Fructose 6-phosphate to glucose 6-phosphate. So remember, that's the first product of glycolysis, and now it's one of the last products, or last intermediates rather, in gluconeogenesis. So you can see how this is kind of like the reverse here. And in the final step, glucose 6-phosphate is made into glucose, and another phosphate is clipped off. Now, another important enzyme is called glucose 6-phosphatase. Okay, so glucose 6-phosphatase, another important enzyme. And this enzyme is found in the endoplasmic reticulum. Okay, the one thing I should point out here right now is that 2 of these reactions do not take place in the cytosol. Remember, in glycolysis rather, everything took place in the cytosol. Nothing took place anywhere else. But in gluconeogenesis, step 1 and step 8 here at the bottom are both going to take place outside of the cytosol. It's not going to take place inside the cytosol. In this case, it's the endoplasmic reticulum. In the case of step 1, I believe it's the mitochondria. So here's a little bit more information that I have about gluconeogenesis. So some more things I just want to point out. So there are many energy considerations, which I already pointed out, being that we need 2 steps in order to overcome the final step of glycolysis. So there's energy considerations that need to be taken into account, and 2 enzymes are needed to get around pyruvate kinase reaction. The first of these enzymes is pyruvate carboxylase and is found in the mitochondria. So there you go. Pyruvate carboxylase is found in the mitochondria. That's the first step of gluconeogenesis. So again, the first step is taking place in the mitochondria. And the last reaction of gluconeogenesis do not occur in the cytoplasm. So I just wanted to say the first reaction and the last reaction do not occur in the cytoplasm. So each reaction that has an energy barrier that needs to be overcome is coupled to the hydrolysis of a triphosphate. So that's the first reaction and the second reaction of gluconeogenesis. They're coupled to a triphosphate, okay? Either ATP or GTP. And notice all of the reactions are doubled. Okay, that's another point I wanted to make. And what I mean by that is that there are 2 pyruvates, 2 ATPs, forming 2 oxaloacetates, a new 4-carbon molecule that was not part of glycolysis. Okay, so what I mean by that is everything's doubled, and I said that earlier in the pathway, that there'll be 2 of everything. And I didn't point out, but I am pointing out now that oxaloacetate is a new molecule that we haven't seen before. It wasn't part of glycolysis, and it's a 4-carbon molecule. It's a 4-carbon molecule, and so it has the addition of 1 carbon compared to the pyruvate. So other molecules can enter the gluconeogenesis pathway, okay, and I mentioned this, I believe, in my other video about lactate. That lactate could be converted to pyruvate by the liver, and that pyruvate could be used to make glucose, and that's exactly what happens there. There's also some amino acids, so if I have the amino acid aspartic acid, okay, you can actually make oxaloacetate from it if you replace the amine group. So you could use amino acids to also form oxaloacetate, an intermediate that would be able to enter the glycolysis, gluconeogenesis pathway. So to make a single glucose molecule starting with pyruvate, you need 6 triphosphates. And the reason you need 6 is because everything's doubled. Okay? So you need 6 triphosphates and 2 NADHs. And the products of glycolysis only produced 2 triphosphates and 2 NADHs. So you can see that it costs a lot more energy for the cell to make glucose than it does to break down glucose. Okay? The products that you make from breakdown of glucose and glycolysis is only 2 triphosphates as a net and 2 NADHs. It requires 6 triphosphates and 2 NADHs, okay, just to make 1 molecule of glucose. So that's quite a bit of energy. And that energy actually has an interesting effect, because if we were to say have glycolysis and gluconeogenesis running at the same time, okay, you would end up just losing 4 triphosphates and get nothing in return. Okay, so if you had both of these pathways going at the same time, one a catabolic and one an anabolic process going at the same time, you would wind up using up 4 triphosphates and get nothing in return. So having both pathways active at the same time would be useless, and it would create what's known as a futile cycle. Okay, and the result of a futile cycle, the result of any futile cycle, is the hydrolysis of ATP, or the hydrolysis of triphosphates, and the production of heat. Okay, nothing else useful comes out of it. It doesn't make sense to break glucose down, right, and make ATP and NADH simply to go back through the process and make glucose again. That makes no sense. Why would you want to do that? And of course, the body doesn't want to waste energy. You know, I always give the analogy that the body's lazy. It doesn't want to do things and use energy that it doesn't need to use. So that's basically why you don't want to create these futile cycles, because they waste energy and they produce heat. Now, of course, there's some instances where maybe a futile cycle's useful, and I could think of one if you're freezing to death, okay, and you need to generate heat or produce heat in order to maintain, you know, homeostasis in the body so that you can survive, okay, but other than that, it's not useful at all to just create heat and waste triphosphates. These are valuable molecules that take a lot of energy, okay, to make. So it simply costs energy, and in order to solve this problem, this is exactly where regulation comes in. Everything needs to be regulated. If it goes unchecked, all these metabolic pathways are functioning at the same time. They're just wasting energy. They're not helping us get to any sort of end. Okay? So we'll move on in the next video to regulation.